Hello guys, it's another good day to learn. Welcome back. So today in hematology, we'll be discussing erythropoiesis. But before we continue, if you've not seen the other video on hematopoiesis, you should do well to check it out because in the course of this video, we'll be referring a lot back to that video on hematopoiesis. Erythrocytes are red blood cells, and the formation of these red blood cells is what we call erythropoiesis. So they have a lifespan of 120 days and are destroyed in the spleen after this period of time. So these cells are produced in the red bone marrow, which are usually found in the epiphysis of long bones. And that's the primary site of formation for all our blood cells, even the white blood cell, the red blood cell, the platelet, and the rest of them. So erythropoiesis is regulated in the kidney by something we call erythropoietin, EPO for short. EPO is a growth factor that is responsible for the process of erythropoiesis. And there are certain conditions that will trigger the formation of these red blood cells. We have conditions like hypoxia. So hypoxia is a condition when tissues do not have adequate supply of oxygen. And when this happens, the kidneys get a signal to secrete EPO in order for erythropoiesis to take place. Also, as a child matures, they require more red blood cells because there is a greater demand for oxygen to carry out metabolic functions. And we all know that the red blood cells are responsible for the transport of oxygen from our lungs to tissues and the transport of carbon dioxide from the tissues to our lungs. Erythropoiesis also occurs as a mechanism to replace blood loss during hemorrhage and anemia. Anemia is a condition of low red blood cell count. So let's quickly see some essential nutrients needed to facilitate this process of red blood cell formation. And those nutrients are vitamin B12, folic acid or vitamin B9, and iron. So iron in its 3+ state is reduced to iron in the 2+ state by a reductase called duodenal cytochrome B, and it's transported to the cell alongside HCl, but it's converted back to iron in the 3+ state before it's released into the bloodstream. So we have folic acid and vitamin B12, and they are both transported from the stomach through the duodenum and finally into the bloodstream. So as we proceed, you will see and you realize how important these substances are to the process of erythropoiesis. So when we discussed hematopoiesis, we were able to establish that blood cells form from a self-renewable pluripotent stem cell called the hematopoietic stem cells. And this stem cell gives rise to two multipotent stem cells called the lymphoid stem cell and the myeloid stem cell. So we know that the lymphoid stem cell is responsible for producing the B and T cells, while the myeloid stem cell, which is trilineage in nature, forms the platelets, the white blood cell, and the red blood cells. And for the sake of this video, we will be focusing on the red blood cells. So we said that the pluripotent stem cell divides into the myeloid and the lymphoid stem cell. So this myeloid stem cell is triggered by EPO, and we said EPO is also called erythropoietin. So EPO will tell this myeloid stem cell that at this point the body needs red blood cell, so the body automatically starts forming red blood cell. And this EPO will reprogram the genes of the myeloid stem cell to start synthesizing hemoglobin for the production of red blood cells. So myeloid stem cell will now become a specific type of cell called the pro-erythroblast. So the EPO will then activate the genes in this cell and they begin to produce RNA. And at this point, the nucleus at the pro-erythroblast stage is very large. It's about 13 to 17 microns, and it divides further to form another cell. So at this point where it divides, the nucleus of the cell is slightly smaller than the initial one. It's about 13 to 15 microns, and the RNA have this blue color. RNA has this blue color, so a reasonable amount of RNA in the cell will give it this blue coloration. So it's called the basophilic erythroblast. And those RNA will keep dividing to form proteins. And what's this protein that it divides into? Here now we have the hemoglobin. Hemoglobin is the protein that the RNA forms. Hemoglobin is red in color, so the cell at this point is already having red color deposits in it. But remember that there are still deposits of RNA which is blue in color. So this cell will have a mixture of red and blue color in it. And that's two colors. So this cell at this point is called polychromatic. Poly means more than one and chromatic is color. That's why we have the polychromatic erythroblast at this stage. The polychromatic erythroblast is smaller than the previous cells, about 11 to 13 microns. And this polychromatic erythroblast will keep converting those deposits of RNA completely into proteins, and the nucleus is drastically smaller at this point. So guys, at the point where chromatic erythroblast is formed, the nucleus is now very small and it's ready to be sent out of the cell. And that's because red blood cells are non-nucleated. They don't have a nucleus. So the nucleus is sent out of the cell alongside the mitochondria. So the macrophages now feed on them. Macrophages are cells that eat up other cells that are not needed in the body or foreign bodies. So what's next? At this point now, a specific cell is now formed and it's called the reticulocyte. This reticulocyte is it's an immature red blood cell, so it remains somewhere in the red bone marrow until it grows into maturation, and when it's matured, it's released into the bloodstream. So you might be asking why the mitochondria had to go with the nucleus. Mitochondria uses oxygen to make up ATP in order to aid the body metabolic processes. And red blood cell, the function of the red blood cell is to transport oxygen from the lungs to tissues and to transport back carbon dioxide from our tissues to the lungs. So if that the mitochondria had remained in the cell, in the red blood cell, it would disrupt the function of the red blood cell. That's why it had to be eliminated alongside the nucleus. So those nutrients we saw, the nutrients we made mention of earlier, they contribute a lot to the whole process of this erythropoiesis, because B12 and folic acid, which is B9, helps in reducing the size of the cell during the formation. If not, these cells will become extremely large, resulting to a condition of macrocytic or megaloblastic anemia. And iron helps in hemoglobin synthesis. So let's quickly have a recap of everything we've discussed. We said erythropoiesis is the formation of red blood cells, and these red blood cells have a lifespan of 120 days, and the main function of the red blood cell is to transport oxygen from the lungs to our tissues and to transport back carbon dioxide from our tissues to the lungs. So we said erythropoiesis is triggered by erythropoietin, which is a growth factor. So the process goes from a myeloid stem cell, which forms the proerythroblast, and moves to the basophilic erythroblast. Then we have the polychromatic erythroblast and down to the orthochromatic erythroblast before the reticulocyte, and then finally we have a matured red blood cell. So guys, if you have any question or you're not clear on any area, do well to leave it in the comment section. Thank you for watching to the end. Don't forget to like, share, subscribe, leave a comment below, and importantly, turn on the notification bell if you have not. Stay curious and keep learning till I come your way next time.